let's look into the electron transport chain and the chemiosmosis. So we know ETC and chemiosmosis happens in the mitochondrial membrane. So let's look into the structure of the mitochondrial membrane. Of the outermost membrane, we have the outer mitochondrial membrane. And the second membrane inside, we have the inner mitochondrial membrane. And between the outer mitochondrial membrane and inner mitochondrial membrane, we have the intermembrane space. Inside of the inner mitochondrial membrane, we have the matrix. In the mitochondrial membrane is the place where the electron transport chain and the chemiosmosis occurs. So let's look into the components that have in the mitochondrial membrane that help in the electron transport chain and the chemiosmosis. So in the membrane, we have the electron carrier protein, which is the complex 1, complex 2, complex 3, and complex 4. So the process is also helped by the coenzyme Q protein and the cytochrome C. In the mitochondrial membrane, we also have the ATP synthase, which will help in the chemiosmosis process. So let's look briefly into the electron transport chain process. So first, the NADH produced from the glycolysis and the Krebs cycle will be oxidized to NAD+, releasing 2 electrons into the complex 1. Of the energy from the 2 electrons will oxidize the complex 1, which will release the protons into the intermembrane space. The 2 electrons will then donated to the coenzyme Q, and the electrons from the coenzyme Q will be donated to the complex 3. In the complex 3, the complex 3 will be oxidized and release protons into the intermembrane space. And the 2 electrons will then reduce the cytochrome, will be donated to the cytochrome C, and the 2 electrons from the cytochrome C will be donated to the complex 4. In the complex 4, will undergo redox reaction, which the oxygen will bind with the protons from the matrix, producing water. And of the electrons transported, the complex 4 will oxidize and releasing protons into the intermembrane space. So that is the first reaction. So the second is FADH, which is produced from the Krebs cycle, and the FADH will be oxidized into the FADH2 will be oxidized to FADH, releasing 2 electrons. The 2 electrons will be then donated to the complex 2. The 2 electrons will then will be donated to the coenzyme Q, and the 2 electrons from the coenzyme Q will be donated to the complex 3. So here the process is most likely the same from of the NADH process. The complex 3 will be oxidized and releasing protons into the intermembrane space. The 2 electrons will then also transported back to the cytochrome C, and the cytochrome C will donate the electrons to the complex 4. And also the oxygen will bind to the protons and to produce water in the matrix. And the complex 4 will oxidize and producing protons into the intermembrane space. Of the transport of the protons into the intermembrane space creates a higher concentration of protons in the intermembrane space. And the higher concentration of the protons in the intermembrane space will create the electrochemical gradient. So a high electrochemical gradient of protons across the mitochondrial inner membrane serves to couple the energy flow of electrons transport to the formation of ATP. So the high concentration of protons in the intermembrane space will create the proton motive force. So the proton motive force will cause the hydrogen to flow back down their gradient through the ATP synthase. So the ATP synthase will use the proton motif force to phosphorylate ADP to forming ATP, which is the energy. So that's basically how the formation of ATP. So together, the electron transport chain and chemiosmosis compose the oxidative phosphorylation. Thank you.
Back to our question given, which part inhibited by the respiratory inhibitor? One of the respiratory inhibitor is rotenone. Rotenone blocks NADH dehydrogenase, which is complex 1 in the respiratory chain, but has no effect on the oxidation of either succinate or ascorbate plus TMPD. TMPD is tetramethylphenylene diamine, an artificial redox mediator which assists the transfer of electron from ascorbate to cytochrome C. Next, the other respiratory inhibitor is anti-myosin A. Anti-myosin A blocks the respiratory change at complex 3 between cytochrome B and cytochrome C. It therefore prevents the oxidation of both NADH and succinate, but has no effect on ascorbate plus TMPD. Last but not least, cyanide is also one of the respiratory inhibitors. Cyanide blocks cytochrome oxidase, which is complex 4, and prevent both coupled and uncoupled respiration with all substrates, including NADH, succinate, and ascorbate plus TMPD.
In conclusion, all these inhibitors will disturb the production of ATP synthase. Firstly, this is because the unstable ratio of NADH and FADH. Secondly, the concentration of hydrogen ion decrease and decrease in number of ATP synthesis. That's all from us. Thank you for watching.